In this first, or A part, of the lecture, we will consider the evolution of quantitative traits in nature, where the form of the experiment is a little bit messier, more continuous than in the breeder's threshold selection scheme. And then in the second, or B part of the lecture, we'll consider some mysteries about the maintenance of large amounts of heritable variation for many quantitative traits. Selection occurs if there is a non-random relationship between phenotypes and fitnesses. But evolution doesn't necessarily occur just because there's selection. It occurs only to the degree that some of that variation was heritable, that is to say, caused by the additive part of the genetic variance. The rate of evolutionary change then depends both on the strength of the selection acting on the phenotypes and the amount of that phenotypic variation that was additive genetic. For directional selection, which is what we'll consider for now, there are two completely equivalent ways to represent this relationship, and this is really the big point of this part of the lecture, is to establish the equivalence between these two ways of writing a prediction of the amount of evolutionary change that will result from selection. The one you've met is the breeder's equation, this one, R equals little h squared times S, which is to say the response to selection is the heritability times the selection differential. And the other way of writing it, more appropriate conceptually for natural selection out in the real world, is as delta x, or delta x bar, just another way of writing R, that is the change in the mean trait value, is equal to V sub A, the additive genetic variance, times this thing beta, which is to say the change is the additive variance times the selection gradient, which is a different thing from the selection differential, but it does much the same job. As we've seen, quantitative traits do often evolve very rapidly. We spent some time looking at the famous beak and body size response of the medium ground finch to the big drought of 1977 that selected really hard for larger beak and body sizes in the population on East Daphne Major. We won't belabor that. You've seen these before. Shows the great increase, which then gradually restored to something like its previous long-term mean body size and beak size because of this trade-off that larger beaks and bodies are favored, really necessary when most seeds are large and hard. But if not, if there are lots of small soft seeds, then smaller beaks and bodies are better because little birds handle those yummy small seeds much more efficiently than large birds do. Okay, so S and beta are both going to quantify the strength of selection. And in this panel, what we're going to do is get at the equivalence between them, even though they look so different. You remember the selection differential is just the difference between the means of the parents who were selected for breeding and all the parents who were potential breeders. That is to say, T star minus T bar for tail length in this made-up hypothetical experiment of selection on tail length in mice. All right, so here's what the selection gradient way of presenting this picture of selection would look like. The gradient is the slope of the regression that describes the relationship between fitness, y, and values of the phenotype, x, when the mean fitness is normalized to 1.0. That is, the average fitness is 1.0. So in the truncation selection case, we have two fitnesses, 0 and something substantial. Okay, this scatter plot made by the authors of this textbook from which I took it, they showed the tail lengths of all the unchosen parents here at a relative fitness of 0, with tail length as the x-axis, and then did the same for the chosen parents. They have a range of tail values, tail lengths, so they spread out along the x-axis. And it turns out their relative fitness is 3. Why is that? It's 3 because they are 1/3 of the population. There were 10 of them and 20 of the shorter-tailed potential parents who, as it happens, didn't breed. Okay, so with 1/3 of them having a fitness of 3 and 2/3 a fitness of 0, the mean fitness is 1. So there's the regression line, that green straight line. That's the selection gradient. It shows a positive relationship between tail length, the x value, and relative fitness, the y value. All right, now here we have our scatter plot again, from which we originally estimated heritability as the slope of the relationship between the average tail length, say, of the parents and the tail lengths of the offspring. This figure is incorrectly labels this mid-offspring value. It's not. It's the individual offspring values. All right. So and here's the regression through the parent-offspring resemblances, and the slope of that, remember, is little h squared. So what we first show with this diagram is that it makes clear why r equals h squared s, or if you like, little h squared is r over s, that is to say the response over the strength of selection, the selection differential, or if you like, the rise over the run. So here I've boxed the selected parents and their offspring. Their joint mean is a point up here on the line. All right, and here we have boxed the unselected parents and their offspring. Their joint mean is a point here on the line. The textbook then shows that idea in this way. O star minus O bar, that is the amount by which the offspring mean moves relative to the parent's mean, is the response to selection. That's what we mean by it in this Breeder's equation context. And P star minus P bar is the change, is the difference between the mid-parent tail lengths, yes, and all of the tail lengths. So that's the selection differential. So that just shows, right, the slope of that line, little h squared, is r over s, or rearranging slightly, the response is the heritability, h squared, the slope of that line, times s. And since we know now that the heritability is the portion of the total phenotypic variance that is additive genetic, we can substitute VA over VP for little h squared and see at once, just as a tautology, that R is VA over VP times S. So VA is a factor of the response to selection. The mouse, upon seeing that, has his aha moment. You see, I get it. The evolutionary response is proportional to the additive genetic variance. But what the mouse didn't quite see was how the other shoe drops. This is a little less obvious, and I won't take time to prove it algebraically, but you can if you're so motivated. It turns out that S over VP is equivalent to beta. It's actually an identity. So then it follows, since there's an S over VP here, may not be obvious, but that's what it is. S is in the numerator, VP is the denominator. So if that's beta, then delta X, same thing as big R, is equal to VA times beta. Okay, the selection gradient has many advantages other than that it can handle continuous and messy relationships between phenotypes and fitness. It also can be extended to multiple dimensions at once, so it allows quantitative geneticists to predict what will happen if two or more quantitative traits are correlated with each other owing to genetic or developmental constraints on the development of the individuals for somehow. And so the textbook concocts this story, which is loosely based on the biology of the medium ground finch, which is that during the drought anyway, fitness goes up as a function of beak depth. That's the one-dimensional selection gradient for beak depth. But they imagine that it tends to go down with beak width. So they're imagining that actually it would be better to have a narrower but deeper beak. But the two are correlated in development, and so what happens at the end of the day is selections pushing in this direction for deeper but narrower beaks, and the population responds along this line because the traits are genetically correlated, and so in fact beak width goes up even though selection favored it to go in the other way, which is a messy reality of real life often, or even of artificial selection. It is also frustrated by genetic correlations often. Okay, an application of this selection gradient idea. Bumblebees select on flower size in alpine sky pilots. Candy Galen noticed that this beautiful flower called the alpine sky pilot, Polemonium viscosum, which grows at tundra elevations on high mountains in the Colorado Rockies, where she works, had flowers that were larger, noticeably larger to her eye than those growing at lower timberline elevations where the trees kind of peter out. Down at timberline, the species is pollinated by a variety of insects, including flies, little solitary bees, and bumblebees. But higher up on the tundra, those smaller pollinators just don't go, apparently, and the only pollinators were bumblebees. And Candy noticed that plants with larger flowers attracted more bumblebees, and flowers that attracted more beads seemed to set more seeds. So she set out to answer the question, could the tundra populations that are not that far away from the tree-line populations have evolved larger flowers because the bumblebees were effectively their only pollinators and preferred and selected for bigger flowers? So she said, all right, I know what I have to estimate. First, the heritability of flower size. It's called, in the jargon, corolla flare. She then caged a large number of plants with bumblebees as the only possible pollinator, allowed them to set seeds, germinated the seeds, planted the seedlings out at random in a natural garden in the natural habitat, and 6 years later counted the survivors. So this is a heroic experiment requiring patience and a great deal of effort to take apart what was happening in nature. So the first part was easy. The heritability of corolla flare turned out to be around a half, a very typical value for quantitative traits. So she could estimate the additive variance the bees had to work with. It would be the heritability times the total phenotypic variance, or a half of 5.66, which is about 2.8. And here are data illustrating where that came from: maternal corolla flare and offspring corolla flare, scatter plot of parents and offspring. Then, from her caged bumblebee experiment, she was able to show that there was a positive selection gradient of 0.13 caused by bumblebee pollination. That is, the average relative fitness of plants increased by 13%, 0.13, per millimeter of corolla flare. The beta has units, which are fitness increased relative to the phenotype phenotype measured in its natural units, which in her case is millimeters. So the predicted response would be, assuming that her experimental conditions reasonably mimic what was going on in nature, delta x in one generation would be VA times beta, meaning something like 2.8 times 0.13, which is a little less than 4/10 of a millimeter per generation, which would be several years. So, doesn't sound like much, 4/10 of a millimeter, but actually it's humongous. It's a 2.6% increase in flower size, diameter, per generation. So a 12% increase could have evolved at that rate in just 5 generations if it was kept up at about the same size and in the same direction for all 5 generations. Here's the data showing how the selection gradient is estimated. It's this faint progression line here with corolla flare of the parent plants on the x-axis, and then relative fitness after this rescaling to make the mean fitness equal to 1. Relative fitness on the y-axis, and there it is, right, the slope of 0.13 per millimeter. Okay, so how fast do quantitative traits generally evolve today in nature? Hendry and Kinnison kind of started this subject really at the turn of the 21st century in a great paper that reviewed 54 different rates of phenotypic evolution that ecologists had estimated in 20 different studies of fish, birds, mammals, lizards, and bugs. They converted all of the estimated rates of phenotypic change to units of phenotypic standard deviations of the trait per generation. The units are called Haldanes, after J.B.S. Haldane, who, as far as anyone knows, was the first person to point out that this was the logical, obvious standardization. And here's a picture of Prof. Haldane holding forth in one of his classes at Cambridge University, I believe, or it might have been University College London, where he taught evolutionary biology for many years in the years immediately after World War II. The median rate of evolution was about 0.03 Haldanes, that is 3% of a standard deviation per generation. Again, this sounds like not very much, but if maintained at that rate, the mean would move by 3 phenotypic standard deviations, which is to say, way out to the thinnest tails of the distribution in only 100 generations, which may be more than we can stick around to observe, but is just a blink of the eye in evolutionary time. Here's a histogram of all those 54 rates, showing that most of them are either effectively 0 or rather small. There's the median 0.03, but a number of them are ridiculously large. In many of those very large ones come from Darwin's finches traits, which I've starred, although there were a few from other systems that were also exceedingly high. Again, here are the units of Haldane's standard deviations per generation. How fast did they evolve over evolutionary time? This is quite a different matter, and the contrast is very interesting. A different scale is used in paleontological studies of morphological evolution. This unit is called Darwin's for some reason. It's the proportional trait value change in units of E, that is the base of the natural logarithms, about 2.8 per million years. And it's easy to calculate. One takes the log, natural log, of the trait at time 1 minus the log of the trait at time 2, that difference divided by the time between time 1 and time 2 in millions of years, and you get Darwin's. Here's a table from Gillespie showing estimates of rates of evolution in Darwin's for different kinds of data that occur on very different timescales. So selection experiments that people had done, Maynard Smith made this compilation, reviewed 8 of them with an average time interval of 3.7 years. They went on at tens of thousands of Darwin's apparent rates of evolution. Recent colonizations, accidental introductions of plants or animals into islands or continents where they hadn't previously naturally existed, have happened. Many of them have happened, and quite a number have been documented with data about trait values and their rates of change. And they tend to produce quite substantial speeds of morphological change on timescales of the order of a century or two, in the hundreds of Darwins. Post-Pleistocene mammals from the fossil record, 46 such cases, with average time intervals of a few thousand years, show net changes on the order of a few Darwins, but that's 2 orders of magnitude less than the recent colonizations. Then fossil vertebrates, going farther back in the recent geological record with time intervals of 1 or 2 million years on average, have net rates of change that are less than 1/10 of a Darwin. So again, down another order of magnitude at least. And similarly for fossil invertebrates, over timescales like of order 10 million years, where the rates are similar to those for fossil vertebrates. So a huge range of rates with scorchingly high rates for short-term evolution, as can be observed by a single human being or several of them in succession or from fossils, and vastly slower overall rates of change over million-year timescales. A couple of years later, Kinnison and Hendry redid their study of microevolutionary rates with a much larger number of traits from a substantial number of studies of 30 different species, so a much bigger data set. Their median rate in Darwin's this time was over 1,000, which is 3 or 4 orders of magnitude greater than the long-term rates of evolution seen in the fossil record. The median rate in Haldane's in this larger, newer data set was about 1/6 of what it was in the 1999 review, around 0.005, but nonetheless, even that is a rate of quantitative trait change that would be vastly higher than what happens over the long term in evolution. So it says there is a whole lot of natural selection happening in the here and now, but evidently it changes direction often enough that long-term rates of change are really very, very small in comparison. So a question I think this raises that I really want to know the answer to is whether many other species besides us will show human-like genomic evidence of rapid recent adaptation, big selective sweeps, and other kinds of soft sweep, but stiff change. And we're now just entering the era when it's going to be possible to get answers for species other than ourselves. And I think this is something to look forward to over the next decade or two. It's going to be very interesting. Okay, so what we know from all these studies of evolution in the here and now is that there is a lot of heritable variation in natural populations. And we'll come back to that in the second part of the lecture. But we want to talk about a way in which that variation is malleable now, that is changeable, which is also a feature of it. And I'm going to start with this famous post-war study by Klaus and Keck and Heise on Achillea yarrow, this composite plant, at different elevations in California where they worked. Here are profiles of 7, I guess it is, genotypes that they featured in one of their classic papers. They wrote long papers that were like small books on this study. So plant height was variable everywhere they looked and heritable in every environment. But in this contrast, which resulted from planting out clones produced by just dividing the rootstocks of different genotypes at 2 of their study sites, Stanford University, just slightly above sea level and near the coast, and Mather, a site at about 4,600 feet elevation in the Sierra Nevada foothills. They found a typical result is that the plants the genotypes did vary, and all genotypes were shorter at the higher elevation. So in 7 out of 7 cases, the Mather plants produced as basically cuttings from the same parent plant, they were always shorter. But some plants that were relatively tall in one environment, take B for example, which is the second tallest at Stanford, it's actually among the shorter half of them at Mather, and so on. So the genetic variance depends on the population's environment, and the environmental variance, which is considerable, depends on the population's genes. That is to say, how much response there was to the environment, to the difference between these two environments. It depended on what genotype you were. Okay? So the environmental variance depended on the population's genes, and the genetic variance was different in each of the environments. Not only the identities of the taller and shorter ones, but just the amount of variance also depended on the environment. So this situation dependence of the variance components, it is called genotype by environment interaction, or G times E, or GXE. And it's important, and it's hard for many people to understand and to grasp the significance of, which is why we're going to spend a few minutes thinking about it. So one way of putting what G by E does is it makes it possible for genetic variation to be created and destroyed by the environment. Now you think of genetic variation being created by mutation, for example, and destroyed perhaps by genetic drift, which is going to reduce the amount of allelic variation present within a gene pool. But this is a different thing altogether. This is genetic variation being created by a change of the environment, holding genotypes constant. And to illustrate this idea, we have to introduce a concept called the norm of reaction, which simply means a graph describing how a given genotype responds to different states of the environment. So that's, for example, what panel A here is. It's norms of reaction for a trait that's modeled on bristle number in larval Drosophila, which is what they're modeled after, as a function of an environmental variable which is taken to be temperature and was in the experiment from which those norms of reaction come. So in this hypothetical case, the two genotypes called G1 and G2, which you can read on the slides, but they're probably not very visible in the video. Sorry. But anyway, that's what they are, two different genotypes. They produce different phenotypes at different temperatures, but always with the same difference between them. They're always different by 2 units of the phenotype score on average. Okay, so this is not G by E interaction. G by E interaction doesn't mean the phenotype depends on the environment. It means different genotypes have different relationships between the environmental variable and the phenotype. These real norms of reaction for bristle number in 10 different Drosophila genotypes raised together in incubators but at different temperatures, they have a lot of G by E, and that's because the lines are not parallel. Some are going up between two temperatures while others go down between the same two temperatures. So in addition to having different means, they have different amounts and direction of change of the phenotype in response to a given change of the environment. That's G by E. So in C and D, which take two of these norms of reaction from the real data, and imagine that the whole world consists just of them, let's say in equal amounts. Genotype 1, half the population. Genotype 2, the other. Now we ask, what in the two different environments, this one with a range of temperatures centered on something like 19 or 20 degrees here in panel C, and then this environment D is the same population of genotypes, the same two, G1 and G2, but in panel D, the environment has a range of temperatures centered on around 24, 25 degrees, which is chosen diabolically to be this point where the two norms of reaction cross each other and have the same mean phenotype at the mean environmental state, that is at the mean temperature. Okay, so what's the difference here between C and D? In panel C, the two genotypes produce distinct phenotype distributions. They don't even overlap, right? The means are hugely different. The ranges are completely distinct. That's because the norms of reaction are, in this range of temperatures, far apart from each other. But in so is there additive genetic variation for the phenotype? Yes. Could we get a response to selection if we selected for a higher or a lower phenotype value? Absolutely. If we selected down, let's say, on the trait value, we would favor genotype 1, so we would get many more of those in the offspring generation and many fewer of genotype 2. So there's a lot of heritability here. But here, the same genotypes, the same two genotypes, in just a different range of temperatures, not that far away from the first range of temperatures, they produce, then, the two genotypes produce phenotype distributions that have essentially the same variance each, but now their means are close to identical. So, if we select for higher or lower bristle number here, will we get a response? No, actually, because the two genotypes will be equally fit given this this distribution of temperatures. And so as long as we keep the population in this environment, we can select till we're blue in the face, and we won't get any significant evolution to happen. We won't be changing the frequencies of the two genotypes. So no additive genetic variation, no heritability. How can that be? Right? We have the same genes, the same two genotypes in environment C and environment D. In one, we have huge heritability of the trait, strong response to selection. Here we have no heritability to speak of, no response to selection. That's genotype by environment interaction, and it happens because the two genotypes have quirky, different ways of responding to differences in the environment. So environmental change created and destroyed genetic variation, heritable genetic variation. So the upshot of this, and many other examples, more realistic, that could be constructed to make the same point, but that would be a bit harder to understand. The upshot is that the heritability of a trait isn't a property of the trait as we conceive and define it. It's a property of a given population, which is to say, roughly, of a given gene pool, in a given environment. And if we change either the gene pool or the environment, we're likely to get a very different estimate of the heritability. In the Clausen-Keck-and-Heise experiment, here are those Achillea plants again, and their genotypes. Height was highly heritable within each environment. The different genotypes produced characteristically taller and shorter plants within each environment. So you could have selected on plant height and got a big response, which would have resulted in changing the relative frequencies of the genotypes. But height in each environment was a poor predictor of height in the others in two different ways. First is that the relative heights differed between the environments, with ones that were relatively tall in one often being relatively short in the other, and so on, even though there's overall a weak correlation of height between the two environments. And the second way in which height was poorly predicted between the environments is that all of the genotypes were substantially taller at Stanford. So differences within populations were largely determined by genes, but with different outcomes in each environment, and the large average difference between populations was entirely non-genetic, entirely a result of the environment, which is to say, the fact that the variation was substantially and unambiguously heritable within each environment and population was not evidence that the difference between the populations and environments must also be a result of differences in genes. There were no differences in genes. This example makes this easy to understand. Both populations were genetically the same, and yet they had this large average difference, but there was huge heritability within each population. It's true, nonetheless, that populations long separated in different environments, in different environments, will adapt to them. And the Achillea genotypes from low and high elevations that had evolved there for a long time did respond oppositely to change. So Clausen-Keck-and-Heise also did an experiment where they fostered or moved plants grown at high altitude to low and low altitude to high, and compared them. So plants from Stanford, from the low altitude population, when grown at a higher altitude, showed big differences. The low altitude population produced many more stems. That's its number of stems in a cluster. What's the word for it? Stems per cutting. The low altitude plants growing in their native environment produced nearly 31 stems at the end of a growing season, whereas the high altitude populations grown at the seemingly favorable low altitude site produced many fewer, right? They were smaller by a factor of 4, actually, or 5. The plants from the high altitude population grown in their native high altitude environment produced 28 stems per cutting. the low altitude plants did relatively better, but still they were worse, right? Significantly worse when grown at high altitude. So, and definitely for both of them, they performed much better in their native environment. Low altitude was better low than high, and high altitude was better high than low. All right, so in summary, there's a lot of phenotypic variation in natural populations, and a lot of it does turn out to be heritable, almost without exception, which is to say, it is possible to select on most quantitative traits and get a response because, at least in most environments, V sub A is in fact greater than 0. It's a fact that not only can populations evolve rapidly, but they actually do in ecological time to a degree that I still think is kind of counterintuitive and surprising. But over evolutionary time, over much longer intervals of time, the net amount of change tends to be rather modest. So how can 2, 3, and 4 all be true at the same time? High heritability, rapid evolution under our noses, but little change over evolutionary time. Well, it implies that environments change frequently and genes flow not too rapidly between populations with different local optima. Otherwise, they wouldn't be able to diverge if there were super high rates of gene flow everywhere all the time throughout a species range. But if it's not too great, then they will tend to maintain lots of genetic variation, much of which will be heritable. And so they'll be able to respond because the evolution, the allele frequency change, won't be swamped by incoming gene flow. G by E interaction, that is to say non-parallel norms of reaction, is probably very common. Every time people have looked for it, they've found it. And it will also tend to slow the loss of genetic variation because the different genotypes are responding differently under different conditions and so on. And that may well also be one of the reasons why there's so much standing variation for most quantitative traits most of the time. Okay, so that's enough of that. We'll come back in part B and talk about these large amounts of variation and why it is sometimes hard to believe they do really persist as well as they